was asked to give a keynote lecture, and I thought, the best is I give a broad overview. So what are the trends in AgBiotech? Because that's actually what I can do best. Diving deep in a keynote lecture and into the science is hopeless because you don't have the time. But maybe drawing the conclusions, what are the trends now and in the future, that's something which is relevant for all of us. It's my opinion, it's my view of the things. It doesn't necessarily have to be the truth. But okay, let's see. Oops, I'm sorry. In brief, I want to tell you what I understand under AgBiotech, because it's not necessarily always the same what people understand here. And so very, very brief back into history, what the first generation of AgBiotech was, what the challenges are, and in the end challenges are also actually drivers for innovation. And so the second generation AgBiotech is something which we have, I think, either on the market right now or in development, either in companies or in research organizations. I also want to make a quick look at biologics, and we will have today, I think, also talks about soil and microbes and mycorrhiza, and that is a very important part, the hidden part. We have to look at this. And well, in the end, for the future trends, it's important to look also at the breakthrough, what I call here breakthrough technologies, which really make things happen, which allow things to happen. And at the very end, look into the future, or try to. So this here, also on the very left, you can see a little time scale. And so this is what I consider AgBiotech. So it starts, on the bottom with the 90s. It starts with the first as a BT and herbicide tolerant things, and then it gets more complicated, multi-gene stacks. That's what we see on the market today, right? Disease resistance, very important, and also tackled through different approaches. And actually on that dotted line, that's where we are right now. And you can see this is a really wide bunch of different traits in AgBiotech on yield. On the other side, on the biological side, I think we are still at the beginning, and much more will come. Okay, so that is, when I say AgBiotech, it includes GM, molecular breeding, actually, because that's also really molecular biology, and in the end, I don't differentiate a lot. Genome editing tools, where in the end, you don't know if the result is a GM or is a non-GM, depends on the state, on the legislature, and also the biologicals. Also I kind of did the following. So when I was at BASF, I was active in that field of yield increase, and of course also on quality, what I call here. Do we have a pointer? I think we don't. But you see more in the middle, a complex quality traits box. So that's why I was active at BASF, and now at Benson Hill, this company is clearly dedicated to yield increase and stress tolerance. So to say, generation 0 of AgBiotech products or GM plants was the Flavor Saver tomato in 1994, which was not at all a success, but commercially, but it was certainly the very first product. And then 1996 on, from that time on, of course, it was Monsanto who was dominating to really bring to the market these first traits. And honestly, I think they happened because it was possible and there was a business opportunity, not so much because the world really was shouting for it. And Monsanto was quite looking forward and saw the needs of the future. And if you see that, it's actually crazy. It's an increase of 100-fold from 1996 to 2013 in the global acreage of GM crops. That's a big success story. And what's actually a little bit scary is the fact that also today, this slide is still correct. Also, it's just on the market. It's just herbicide tolerance and insect resistance. Single gene trait, quite simple, because there's not much interference with the plant, the crop metabolism itself. So that is, of course, today you have triple, I think in the meantime, even at least in development, quadruple stacks of herbicide tolerance and BT to make them even better. But in the end, it's only an optimization of an existing theme. And well, in the major crops, the penetration rate of GM is really high. If you look at soybean, and these numbers are from 2012, I didn't find newer ones. So I think in the meantime, soybean is beyond 85% globally, and corn is also much higher than 32%. So it's quite amazing. Cotton, of course, is essentially GM wherever you go. Sorry, we should also have a little look at these potentially on the right side on these future biotech crops, which are well, look here, no? Also, in China, there is GM rice actually waiting to be approved. It's just a matter of maybe the political environment and the right point in time that it happens. The challenge is also an innovation driver, as I said at the beginning. So the challenge is really because the world population is growing so much, because people are getting more wealthy and the middle class is growing. Look at the bicycles in Beijing 10 years ago, 15 years ago. You saw many more bicycles than now. Now they're all driving cars, which is good in a way. Well, it's a matter of point of view. But so growing middle class means eating more meat, and in the end you need more agricultural areas. Very limiting, doubling productivity is a challenge and an innovation driver, as I said. And the first of the second generation AgBiotech traits I wanted to talk about is disease resistance. I don't see a big trend here because it has been worked on. The big companies have worked on this in the 90s already. Look at wheat. Monsanto and Syngenta both had wheat GM programs in the 90s and shelved it because they thought there's no acceptance of GM wheat. Let's keep it there. That's why also most fungal disease resistance programs are non-GM, just through conventional and advanced breeding. BASF, where I was, actually they had a great product candidate, Phytophthora-resistant potato. It's a big disease and it's really important, and the product was great. But it was really working well. In the end, it was shelved. After it was approved, it was not put on the market because the major market was in Europe and Europe didn't want it. In development, at the big companies are GM soybean, which is resistant against Asian soybean rust. Of course, also a little bit earlier in development, disease-resistant corn plants, root, stalk, and ear rot resistant corn. So that was the disease resistance part. I mentioned where I was at BASF in the early days, complex quality traits. What I say here, what I mean is not improving performance, but improving the nutritional factors, the quality of the crop. And we all know the golden rice story, which started by research institutes, and also Syngenta actually helped a lot to make this humanitarian project to increase beta-carotene content in rice a success. And now it's Erie, which is really bringing this, further developing it and bringing it on the market. But it was started in the early 90s, and now it's 2015. This is very long. This is too long. Eh? And same thing, even the big companies can't do it much better. Look at my project where I was when I was at BASF, as a long-chain polyunsaturated fatty acid production in canola, in rapeseed. Very healthy fatty acids, kind of fish oil in rapeseed. That was cutting-edge research, but it also means, and it's really establishing a whole metabolic pathway into this oilseed crop, which involves 8 genes, 8 gene promoter combinations, and a lot of research behind it. This is now close to commercialization together with a partner, Cargill, but it is still not on the market, and it also was started before 2000. That's 16 years. Not good, because it is much more complicated than originally anticipated, right? Because there's a lot of interaction between that metabolic pathway and the crop metabolism. And also the tools which you have available today were not really well established then. So metabolic engineering was there, but not of complex traits. synthetic biology, not really. So I also should note that most other GM approaches about quality traits failed. Monsanto did it, BASF as well, Syngenta also, for example, trying to improve the nutritional composition of corn, improving the fatty acid composition, the protein content of corn. Technically it worked, but in the end the value chain didn't want it, and these products never made it to the market, although they were advanced. Yeah, it's more than just science. The market has to be there. And okay, so the biggest opportunity and also challenge is to increase yield and stress tolerance in crops. This is taken from a Monsanto presentation where you see the market opportunity if you increase crop yield or yield stability. Because in the end it's in the field. In the field there's always stress, as we know, all kinds of stresses, and in the end it's what comes out. That is yield stability and yield increase. Oh, sorry. Okay, let's go here. And so here are, of course, again, based on molecular breeding, products from Pioneer, from Syngenta, and from Erie, and from many, many more organizations, right? So Aquamax and Artesian, these are both corn, which really have, without GM, very nice, optimized drought tolerance performance, right? Same with Erie, also there's also a submergence tolerant rice. This is all non-GM. And the transgenic approach was taken by Monsanto and BASF, and the drought guard is on the market already since little more than 2 years now, on a large scale since 1 year. And this is, in the end, a bacterial RNA chaperone, which helps to make, to stabilize the corn RNAs actually under drought conditions, and it really works in the field. That's a big investment which these both companies did, and it's a very high throughput screening. They really screen a lot of candidate genes, bring them into the crops, into the fields, test them there. Big effort, and not everybody has $2.5 billion to run this machinery. It's actually a little crazy, yeah. And so what Benson Hill does now is on a much smaller scale, very much focused on yield improvement through improved photosynthesis. This is just one aspect, but we believe it's very important. I can tell more about this tomorrow in the other presentation I give. Now let's look under the soil. Or not all of these guys are under the soil, but at least they're very tiny, the biologicals, which are usually bacteria. And this is actually the most amazing and rising aspect of AgBiotech, which I consider AgBiotech. This growth of that market volume and on the capital which was invested in it is very amazing. As if you look at this, in 2012, which is just 3 years ago, it was 1.6 billion globally. In a year from now, it's supposed to be doubling. This is quite impressive. Also, if you look a few years back, how many small companies were acquired by big companies, and how much money was spent there for Athenix, AgraQuest, Pastoria, Becker Underwood, and also this big alliance between Monsanto and Novozymes, which is existing in the area, is actually a little bit crazy, because in the end they are competing with agrochemicals, which are very well established, and they have to be as well working as these chemicals. I think very important independent players today are AgBiome, Bioconsortia, New Leaf Symbiotics, and very young, also promising Growscientia. All of these 4 companies are in the US. I'm sorry, there are maybe much more and also important companies elsewhere. This is my point of view. I'm sure it's not complete. And a little bit abroad, but it's considered also as biologicals, is Monsanto's sprayable RNAi, what they call BioDirect. It's, I think, a little different, because in the end it's like a chemical which is sprayed, but it's a biological because it's RNA. You know what I'm talking about? It really works well. Also you have to produce the RNA, which is in the end RNAi, and either controls weeds or insects or diseases. It's, I think it was shown by Monsanto that it works well. I think the challenge here is and will be to produce it cheap enough so that it really can be applied on a large scale, cheap enough and stable enough. in the field. So I mentioned at the beginning, which are the breakthrough technologies which actually enabled this all. Obviously, sequencing costs went far down by a factor of 10,000. And honestly, I know this is nothing new to all of us, but that really enabled all the other things which come now. Because if you had to pay $100 million per genome today, then you would not do all the molecular breeding and genome genotyping by sequencing, which you can do today. So here, really enabled by this, I call it sequencing revolution, is all aspects of molecular breeding, genotyping by sequencing. Also the new RNA-seq experiments, which are so helpful for transcriptomics, are not possible without it. Genomic selection is, again, also based on sequencing, an amazing mix of genomics tools and also computational and algorithms where you can really predict the phenotypes before you actually see them based on the genome sequences of the parents. I find it really amazing. And Benson Hill is also doing this, applying this to our platform now. Interesting here also, let's look at the animals. For example, genomic selection is much more advanced in cattle than it is in crops. Also much more. We can certainly learn from there. So if somebody wants to be on the forefront, hire somebody from the cattle industry who did genomic selection there. Okay. Phenotyping, or looking at the phenotype of the crop. This is also something which really enabled what we see today. And looking at the research, and this is a research conference, of course, it started all under well-controlled conditions, so indoors growing of the crops, and crop design, which then was acquired by BASF, was certainly leading in that field of automated greenhouses, which have all kind of imaging systems, not just visual, but also other sensors, and really monitor these different, rice plants in this case, which have different genotypes, and looks at the phenotypes, right? Of course, the other companies actually did similar things, not in the same, but similar manner. Monsanto Syngenta Pioneer certainly did that. You may know in Europe there is this Keygene LambdaTech kind of joint venture, PhenoLab, which also provides something like this. In the academic world, I think what the Donald Danforth Plant Science Center in St. Louis has is very impressive. It's a lambda-based, but not purely lambda-tech-based system. And also very advanced in his individual field. What David Kramer at Michigan State University does on photosynthetic phenometrics is very impressive. Very controlled conditions. So the next step is then to do this outdoors. And CSIRO, also in Australia, they have this, they call it Phenomobile platform, which you can see on the right, which is in the end sensors attached through something which goes through the field. And you can do it in different ways. I think also Huazhong Agricultural University in Wuhan also has a nice phenotyping system. I didn't see it yet, but I will soon. University of Nebraska-Lincoln also wants to install something like phenotyping in the field with spider cams, which are flying, where the sensors are sitting on spider cams, which are then flying above the crops growing. Or you can do it the other way around. As you can have, and really, I mean, sensors are getting cheaper and cheaper, so you can do this. You can install sensors, cameras in the field, or you move the cameras and fly above the fields with drones, either fixed wing, multi-copters, and it's really amazing. And here I want to point out particularly hyperspectral imaging, how much information you can extract from hyperspectral images if you have the right ground truthing data, and you can really quantify the signatures you get to water content, nitrogen content, chlorophyll, photosynthetic activities. It's amazing. Environmental data, weather data, soil data, very important. And then we will hear a lot about soil parameters later today. Soil mapping is really important. pH, electric conductivity, organic matters, and so on. That really determines the yield potential in the field. And big data integration, I'm not the expert, so I know it's very important, but I really can't tell you where to look at this, where to go to if you look at this point. Certainly there's Climate Corp, which was acquired by Monsanto, but there are many others. Sorry. Okay, I indicated it at the beginning, so genome editing is very important and is really something which came up very recently. We know the zinc finger nucleases from Sangamo, which were then working with Dow AgroSciences, meganucleases, and TALENs as the more recent technology. They all work with different principles, and the latest in the row here is, so to say, CRISPR-Cas, which is a little different to the others in a way that it's RNA mediated. I would say it was the technology of the year last year. I think it was 2014, and it went into 2015. The number of journals and publications and patents on that technology, amazing. And it's very versatile, precise. I think the IP portfolio and landscape is still very open and very inconclusive. That is a little scary to big companies, but it's also an opportunity for smaller companies and for all kinds of research activities. There seems to be one player in the plant field. It's now called Calyxt, which used to be Selectis Plant Sciences, actually, the plant science part of Selectis, which seems to acquire a few basic patents in that field. Actually a shame. I wish it would be more open so that it can be really used more broadly without dependencies on one player. Yeah, and really, CRISPR-Cas, I mentioned it's versatile. Really what you can do is not just precision, as a precise integration into the genome. You can delete precisely, you can also knock out precisely, but you can also activate and deactivate. No? With the dCas9 subtype of the CRISPR-Cas technology package, you can actually do this. It is really very impressive. I'm sorry. Okay, so very important for the future, for precision crops. And one more thought about the future. Now we are looking more forward. Also, these are grain bins, but I consider these silos. And honestly, a lot of the activity of the research in our field has been in silos, and that's never good if you don't work together with the others. Also, if you look at I didn't really mention advanced mutagenesis, but I mentioned genome editing, phenotyping, genomic selections. I mean, the big players, also Monsanto certainly has several of these things in their organization, but it's a huge organization, and that makes it again slow. Smaller companies often provide only different aspects, and also research institutes don't really look at the entire portfolio, but I think if you do it, and if you also have more integrated approaches, you really increase the innovation potential, and it's not impossible. You don't need 200 computational biologists like Monsanto has in one place to handle precision agriculture. I think it can be done with a smaller task force. So if I look at the future, these integrated discovery platforms are certainly key, and I'll tell a little bit more about how Benson Hill is approaching this. Precision crops, also where really you know where you integrate your transgene or you up and down regulate it. And I didn't mention this, but we and also others have USDA approval letters that approaches using genome editing are considered non-GM. If you start from a crop gene and you end up in a crop, you can do it easily without being a GM, a genetically modified, having a GM product. Now looking at the biologicals area, what I think the future is here to study more the crop microbiome interactions. And there are hopefully good university groups. The company which really does that extremely well and innovatively is Bioconsortia in the US, who really looks at consortia of microbes, what effect they have, then they select those teams or the consortia of microbes which really have the effect, and they, at least they say, they do this along with the breeding program of the crops. So this really means here are not two silos, but they are actually working together, the biologicals and the crop breeding. part. Also important for biologicals, reality is, I come from BASF, a biologicals product is nice, but in the end you combine it with chemicals because there is good chemistry out there, and that's why formulation is very important. It must be possible to combine the chemistry with the biologicals, or to have the right time schedule when to stop with the chemistry, when to apply the biologicals when it comes to harvesting and so on. These are things which are not solved yet. So if you look back to that picture which I had first, and now I went a little bit more ahead until 2030, I really see these yield increase precision crops where you really know exactly where your transgenes or your modifications end up. Quality improved precision crops, of course, and also on the biological side, integrated products for improved crop microbiome interactions as the longer perspective trends in AgBiotech. So thank you.